Okay, so we will be presenting on Spirobranchus giganteus, which is also commonly called the Christmas tree worm. We have Colin, which is going to be doing over the life cycle of distribution. Sadie will be covering ecological importance, predator and prey, and I will be going over some taxonomy and morphology of the Spirobranchus giganteus annelids. Okay, to start off, we're going to be covering a little bit of taxonomy. Spirobranchus giganteus is part of the Animalia kingdom, phylum Annelidia, class Polychaetae. Although about 5 years ago, as Lockwood said in one of the previous lectures, they are undergoing some reclassification as far as taxonomy is to whether or not they are polychaetes. Don't call them oligochaetes, and you'll be good. They are order Sabellidae, family Serpulidae, genus Spirobranchus giganteus, and they were first described by Pallas in 1766. They include 20 different species, according to Tenhove 1997. However, class Polychaetae in particular have undergone, like I said, some recent taxonomic changes. According to Tenhove in 1997, Spirobranchus is a complex composed of 3 different subspecies, which is actually dependent on their geographical location. So you have giganteus, which is limited to Greater Caribbean and Mesoamerican Barrier Reef, and then you have Incrustatus and then the Spinosa subspecies. So it is possible that because Spirobranchus is subtropical and found in various parts of the world, that you can actually get 3 different subspecies through isolated breeding and speciation events. So in other words, like if you have subspecies A over here in the Greater Caribbean and then subspecies B over here in the Great Barrier Reef, they may not be able to breed and share those genes, so you end up with subspecies in different areas. Okay, going over a little bit about the morphology. This right here is actually the general body plan of the tube-dwelling annelid. The prostomium is the only external visible part of the organism, which can actually grow to anywhere between 2.5 to 3.8 centimeters, which is about the equivalent of 1 to 1.5 inches, according to DeMartini 2011. These are marked by brightly colored whorls containing pinnules, which is used for their feeding strategy. They are filter feeders. The pinnules are arranged in a tight formation, as you can see right here. So whenever they actually move and they beat together, they create a water circulation, which helps filter out the detritus and phytoplankton and their particulates that they feed on. So according to DeMartini 2011, their Tenhove 2011, according to Tenhove, their collarchaetae and operculum shape help differentiate the different species within the Spirobranchus genus. They are segmented, which means that they do exhibit metamerism and repeating segments. Their anus and their mouth are actually located at the opening of the organism right next to each other, according to Tenhove 2011. They do secrete calcium carbonate, which makes up their tube. Their tube can actually grow to be about 20 centimeters, which is approximately 7 inches in length, and their tube is actually dependent on their age. So the parapodia are absent in Spirobranchus giganteus due to their sessile nature. However, they do have chaetae for anchoring within the coral substrate, which is this structure right here. So this is the actual operculum, which is actually used to, whenever they suck down into their hole, the operculum will close to help protect the organism. And I'm going to be handing this over to Sadie, and she will go over some ecology and predator-prey issues.
Hi guys, it's Sadie. I'm going to start off with the predator-prey relationships of organisms in accordance to the Christmas tree worms. Christmas tree worms are filter feeders via the cilia on their radials, which we talked about in the previous slide on morphology. Their primary food sources are zooplankton, phytoplankton, and detritus particles, according to Toonen 2012. The predators of these types of worms are crabs, shrimp, sea urchins, and some tropical fish, primarily the butterfly fish, as we have pictured here. This is according to privatescuba.com. Copepods are actually a parasite to the Christmas tree worms, according to Tenhove 1994, which we will get into the importance of that here in a minute. There are no known diseases for the worms. So now for the ecological importance of its mutualistic relationship with coral. They recycle detritus and organic materials according to Hutchings 1983. The worms themselves shield coral species polyps against predation, primarily from the crown of thorns sea star Acanthaster plansii, according to Devantier et al. 1986. The coral themselves house the larvae and adults, so that's great protection for the worms against their own predators, according to Smith 1985. There has been seen in research an improvement in waste product dispersal, according to McCady et al. 1998. There has been an increased nutrient availability from waste, also according to McCady et al. 1998. There has also been seen in research there's an increased coral growth in size, eutrophication, and circulation and production, according to Glenn 2013. With the mutualistic relationships with these worms and the coral, Christmas tree worms have been found to have longevities between 10 to 40 years while living on coral porites, according to Nishi and Nishihira, 1996 and 1999. It had also been found in another research article called from Yap et al. 2009 that if the coral dies, the smaller sized worms will likely die, whereas larger worms can carry on. And this is because they saw a comparison between the dead and living porites. The smaller worms were absent with dead porites, and they theorized maybe this has something to do with algal overgrowth, but it's unconfirmed. So now for just the general ecological importance, relationship with others. Small crustaceans seek refuge near the operculum spines, according to Yap et al. 2009. This is actually where the copepods and amnipods are housed. And like I said in the previous slide, the copepods are actually a parasite. And this is found in Yap et al. 2009 and Tenhove 1994. They are biggest in being indicators of reef health, according to Yap et al. In 2009. They facilitate fast recovery of coral against bleaching events, predation, and overgrowth of turf algae, according to Heathcote 2007. And larger corals and more worms seen in the environment usually indicates that there's less ecological disturbance in that specific area, according to Yap et al. 2009. It also has been theorized that smaller corals may not attract as many worms. The 3 spots were actually in another study, according to Tuzzi v. et al. 2006. 3 spots where coral still lived with worms, the rest was dead. 1 year later, the coral was growing back, though thought that the worms living within the skeleton improved water circulation that decreased susceptibility to coral bleaching. So, like I said in the previous slide, they're very important for the circulation of the water and the particles and everything. So it's basically like they're managing the coral. So they're very important. And this was also shown in Nakamura et al. 2003. And I'm going to turn it over to Colin now, and she's going to talk about the life cycle and distribution range of these species.
Hey guys, I'm going to talk to you today about the life cycle and reproduction and distribution of these organisms. So, Spirobranchus giganteus is going to be gonochoristic, which means that both male and female parts or sexes are going to be on separate organisms. They're also considered broadcast spawners. That means that they release the gametes into the water and they're fertilized externally in the water. And I also found some research off of the Gulf of Mexico, which observed that Spirobranchus giganteus sometimes spawn simultaneously with their host corals. They found that this normally occurs during a neap tide, which is right after a new moon. And that also occurs with little wind or wave action. And what they suggested with this simultaneous spawning is that the coral larvae and the Spirobranchus larvae, they tend to settle at the same time in the same specific location over the corals where the adults were actually still living. Along with part of the life cycle, we also have the larval growth stages. And within 24 hours after fertilization, one of the first things that develops is a larval ocelli that is considered like a photoreceptor. It's described by Smith, 1984, A and B. It's used for light responses until the branchial photoreceptors are more developed after settlement. Within 1 to 4 days, it's considered a trochophore larvae. It has a band of cilia that is used for locomotion. This band or ring of cilia moves in opposing directions, and it's used for movement or swimming. At 4 to 12 days, S. giganteus larvae begin a searching phase. And it's usually approximately at 12 days that they begin this. And this behavior is thought to be by, or found or suggested by Marsden in 1986, is thought to be the development of coral species recognition. After settlement, Spirobranchus giganteus secretes a mucus tube at first. This is the basis for the calcareous tube. As stated before, the larvae of S. giganteus does not bore into the coral, rather the larvae build a tube that is incorporated into the new growth of the coral skeleton. And that's described by Rowley 2008, Smith 1984A, 1985, and Roos and Pilelli 2001. Spirobranchus giganteus' distribution is circumtropical. It's found in the Atlantic, Caribbean, Red Sea, and Central Pacific. I would consider that equatorial. If you look at the figure on the right, you can see that it's a map of the world distribution of Spirobranchus giganteus, and you can kind of see that it goes along the middle equatorial line of the map. Along with where they're found geographically, prevalence is also dependent on the coral species available, so that different species or subspecies are going to prefer different types of coral host according to where they are. Spirobranchus giganteus actively select coral hosts, and that's found by Hunt et al. 1990. They found that S. giganteus prefers massive coral species, and Dai and Yang 1995 proposed that this might be that massive species of corals have a longer lifespan. With also in consideration of that, we found that mean worm density is greatest at 6 to 18 meters in depth. So you'll see the greatest aggregation of these worms on corals at 6 to 18 meters in depth.
Okay, so that concludes the presentation. If you guys have any questions, feel free to ask us.
Bye, guys.
Thank you. Bye.